Welcome to the second talk of our morning series here at Saturday University. Today we're going to hear from Kirsten Kapp. Her work is focused on human-environment interactions while pursuing her master's degree at the University of Wisconsin at Madison, the other UW. She worked on human and black bear conflicts in northern Wisconsin. And more recently, she's shifted her focus to the question of how human waste, particularly that of plastics, impacts the environment and the animals in it. We've all seen the pictures of gulls caught in the plastic rings of six-packs or of seals caught in plastic fishing nets. But Kirsten's interests lie in smaller plastics, the almost unnoticed microplastics, and how they are ingested by fish once they enter the environment. At the moment, she researches the herring off the coasts of Maine, a project sponsored by INBRE, a research grant organization sponsored both by the University of Wyoming and the National Institutes of Health. And at CWC, where she has taught since 2006, Kirsten has worked to bring such research experience to students, introducing them not just to knowledge, but how that knowledge is discovered and formed. Today we'll hear about a project that has been supported by the Teton Conservation District, in which Kirsten takes her class of students out into the field to study microplastic pollution in the Snake River Watershed and its impact on the water quality out of that watershed. Kirsten.
Thank you, Paul, for the introduction. And I also want to thank everybody with the University of Wyoming that have brought this amazing lecture series to Jackson. I also want to thank the Center for the Arts, Central Wyoming College, Wyoming Humanities, and Think Wyoming, and the organizers for the amazing and awesome Wild Science Festival that's going on right now. I am really excited to be here today, and I'm often awestruck at how excited I can get to talk trash. And I'm not really sure how I got from wandering around the woods chasing charismatic megafauna to looking under a microscope at bits of trash, but I find it really exciting for a number of different reasons, and I hope that I can inspire you and make you think about this potential devastating problem today as well. So I think that the best place to start is to go back in time. And what I'm going to do is I'm going to tell you a story. I'm going to tell you a story about our plastic footprint, the history of plastic and plastic productions, and then I'm going to move on and talk about what the potential ecological consequences of that plastic pollution are. And finally, I'm going to describe to you what students and I are currently working on as far as looking at microplastic pollutions in the Snake River. So if we take a step and go back in time, the quest for materials such as plastic is tied directly to the fate of the elephant. And back in the mid-1800s, and unfortunately as they are still today, elephants were being brutally poached for their ivory, for objects such as toys and trinkets, combs, piano keys, and the billiard ball. It turns out that the billiard ball, or the pool ball, made of ivory was unmatched at the time. It had the perfect roll, the best rebound, the greatest bounce, and there was no other material, metal, wood, ceramics, that could match that. And back in the mid-1800s, billiard was a really popular form of recreation. And so there was an increased demand for pool balls and pool tables. In billiard halls, for example, in Chicago, about this time, 850 billiard halls. Today, there's less than 10, to give you an example of that. But with the quest for these perfectly performing pool balls, ivory was becoming more and more challenging to come by. It was getting more and more expensive. It would take 2 elephants to really make one set of pool balls. You could get maybe 3 or 4 pool balls out of one ivory tusk. If that, you might have to, or of that, you might have to throw away one due to imperfections. And then, after you make them, they'd have to sit on the shelf for up to 2 years before you could actually play with them. Talk about a lack of instant gratification. So a man by the name of Michael Phelan, who back then we termed the father of pool, he was one of the major billiard suppliers. He recognized all these challenges associated with finding ivory to supply this increasing demand for pool balls, and he put an ad in the paper. So he put this ad in 1863 for a financial award, and back then the value was $10,000. So that's a pretty significant financial award. And he was going to give it to the person that could create a material, such as ivory, that would then match ivory in pool balls. And there were a few attempts. The first attempt was by a man by the name of John Hyatt, and he made a product called celluloid out of plant-based materials. And celluloid, although it did have many uses, it didn't take the cake. He didn't win the prize, although he did actually make pool balls with it. He coated pool balls or plaster pool balls with this celluloid product. It did replace ivy in piano keys. In fact, you could have a choice if you wanted piano keys with a celluloid coating or an ivory coating. So it was fairly successful, but just not in pool. In fact, it had a tendency to ignite. And so if you're playing pool in a billiard hall, the last thing you want is to hit a pool ball and have it spontaneously ignite and catch on fire. And the celluloid actually was successful in creating film strips that were used in theaters and cinema. And because of its igniting capabilities, did actually cause a few fires. Well, that was in 1868. So if we fast forward, the next invention that really matched ivory in pool balls wasn't until 1907. And this was by a man by the name of Leo Baekeland. And Leo, at the time, was experimenting with resins. And particularly, he was experimenting with phenols from coal tar and formaldehyde. And what he was doing is his goal was to increase the strength properties of wood. So he would inject this product into wood to make it stronger. And during one of his lab experiments, by accident, he created this product that matched ivory and eventually replaced ivory pool balls. And he called that product Bakelite. And he advertised that product to the public at the American Chemistry Society meetings in 1909. And Bakelite took off. Not only could it make amazing pool balls, which had just as good roll, bounce, and rebound as ivory, but we see Bakelite buttons and sunglasses and phones and radios. So they called it a material of 1,000 uses. And ironically enough, the logo that they had, or that they decided to use for their product of Bakelite, was the mathematical sign for infinity. So up until this time, though, from 1907 until 1930, they were developing more and more of these plastic products, and it really inspired others to develop other types of plastics. But during the time, there was a lot of these designs caught up in patent war. So the plastics weren't really available for the general consumer, even though there was a huge demand at the time for it. And then we had World War II. And during World War II, all the plastics that were being produced, or all the energy being put into plastic production, was for making products that were used in the war. And then when the war ended, plastics hit the market hard. And we fell hard for plastics. We were lovesick, as we still are, for their durability and their versatility. Right? Plastics are amazing. We have made many products that benefit society greatly. Pacemakers, artificial limbs, solar panels, lighter and lighter vehicles to get more or better fuel efficiency, right? And let's face it, ski goggles and ski boots, right? Plastics, okay? And so plastics really, the production of plastics really dramatically increased since then. And we entered 1955. So some of you may have seen this famous photograph. It was the cover of a 1955 issue in Life magazine. And we entered this era of throwaway living that we're still very much living in today. And the article that accompanies this photograph, the first sentence of it reads, The objects flying through the air would take 40 hours to clean, except that no housewife need bother. They are all meant to be thrown away after use. And that's really where the problem lies, right? The throwaway part. Back then, and very much still today, we don't think about where away is. It's broken, just throw it away. Yeah, throw it away, we'll got to take a run to the dump or to the landfill. Okay, and that's really where the problem starts. So with the increasing plastic production, this is a graph from the Ellen MacArthur Foundation showing this dramatic increase in plastic production and the amount of plastic produced per year. 2014, we reached 311 million tons. That is estimated to increase 4% every year. Okay? And again, I mentioned the societal benefits of that. But part of the problem is in mismanaged waste programs, especially in developing countries. A lot of this plastic is actually ending up in our oceans. It's really hard for us to estimate and calculate how much plastic that is. But in a study that was just published last year in Science magazine, scientists attempted to estimate what that was. And they calculate that on average every year, 8 million tons of plastic are entering our oceans. So that's a lot of plastic. So if we look at the marine debris research that follows this plastic production, this is a bar chart, and you can see the pattern correlates quite nicely with plastic production. This was actually from a book that was just published last year called Marine Anthropogenic Litter. If any of you are interested, it's an all-encompassing book that looks at the history of marine debris research. And as we increase production of plastic, more and more ends up in the ocean. And you can see back here in 1965, we have some of the first accounts that are published in peer-reviewed scientific literature. So I must note that there were accounts made of debris in our oceans prior to this. Okay. As we move further up, 1970s, you can see the blue here is research looking at amounts and sources of marine debris. The green then correlates to the first impacts we start to see of marine debris on our wildlife, and that would be ingestion. We also see entanglement, which Paul mentioned in his introduction, those photographs we've all seen that are fairly disturbing to look at of gulls and seals being entangled in marine debris. And then you can see these categories, kind of the literature in each one expands as we move up in the time frame. And then we even get to some policy. So in the scientific community, the interest of marine debris started to really kind of take hold, and there were some conferences dedicated to talking about marine debris. And then we have this wane here, okay, kind of a lack of publication. Now, I'm not really sure how to explain that. It could have been a lack of interest. Sometimes it's explained that these initial accounts are really just kind of out of mere curiosity. But we do see a resurgence here happen. And we owe the resurgence in marine debris research really to just one man. And that man is Captain Charles Moore. Captain Charles Moore is a retired Navy captain and avid sailor. And in 1997, he set sail, he raced on what's called the Transpac Regatta. It's a well-known historical sailing race from Los Angeles to Honolulu, Hawaii. And so he and his crew sailed the race, they ended the race, and they had a lot of fuel left over. Said, well, let's just cut through the gyre. Okay, now in a minute I'll talk about what a gyre is, but let's just cut through the gyre on the way home. We have plenty of fuel, we've never been in that area of the ocean, let's do it. And when they did, Captain Moore was astounded by what he saw. And in an article he wrote later in Nature magazine in 2003, he recounts his first observations sailing through the gyre as, no matter what time of day I looked, plastic debris was floating everywhere. Bottles, bottle caps, wrappers, and fragments. And this is a photograph of, let me go back, apologize for that. This is a photograph here of him holding a water sample that he collected, and you can see all these plastic fragments that he speaks of. And along with his recounts of his adventure through the gyre, the media took hold. So how many of you have actually heard of the Great Pacific Garbage Patch? Yeah, we owe that in thanks to the media campaign, right? And although the media have falsified the description a little bit, that's actually okay because it really grabbed the attention of the community. And so I'm just going to pause for a second and say, well, what is an ocean gyre and how does it form? And I'm going to try to explain why Captain Moore was seeing garbage in the areas of the ocean that he described. And this is a still image from actually a computer simulation that was just published last year as well. So I'm going to kind of emphasize this was just published last year, that's published this year, because there's a lot of amazing work happening right now. And this data, so if you can see the difference between these little white specks and then blue specks, and there's a lot more blue specks than white specks, that's because the blue specks is simulated data, so simulated data points, based on the real data, which are the white specks. And these are drifter buoys, data from drifter buoys. So what these two computer models did is they looked at data from 30 years of drifter buoys. Now, drifter buoys are buoys that you set adrift in the ocean, but they're often equipped with satellite transmitters or GPS units, and then perhaps temperature probes or other probes that you'll use to collect scientific data. And they followed where those drifter buoys ended up in the ocean. And based on wind patterns and ocean currents, okay, they ended up in the same areas of the ocean where we see our marine debris. So seaweed, marine debris, plankton, all tend to accumulate in what we call gyres. Right now we know of 5 gyres, although we're thinking there's probably a 6th up in the Arctic, in the Barents Sea of the Arctic. But with these gyres, these are the areas we are seeing a large accumulation of debris. Now, I'm going to take a step back for a minute and say, okay, well, the media accounts were a bit falsified, right? They advertised as this great Pacific garbage patch, the size of Texas, like an island of trash. Well, it couldn't be further from the truth, okay? There's no island of yogurt containers and water bottles bobbing up and down in laundry baskets, and you certainly can't get out and walk across it, okay? These are actual photographs from the Pacific Gyre. A marine biologist by the name of Miriam Goldstein, who spent her whole PhD studying marine debris in the gyre, took these photographs. And so while you might see a large item of floating debris as you pass through the gyre, more often what you're seeing are these plastic fragments, still signs of anthropogenic activity. And when I say marine debris, I'm kind of using an umbrella term. Marine debris defined is any man-made floating object found in a body of water. But as we do more and more research and collect more surveys on the trash that we're finding, we're realizing that the majority of marine debris is actually plastic. So this is data here from 608 beach surveys across 8 different countries. Okay, now the data is a little outdated now. It's from 2007, but the trend is the same, in that an overwhelming majority of the debris that we find, 75% this study reports, is plastic. Other studies report up to 95% of the debris that they find is plastic. And plastic comes in many different shapes, sizes, and from a lot of different sources. So up here on the left is a photograph that I took after serving as a guest scientist on a sailboat, a research boat called the Rozalia project off the coast of Maine. This is actually Hurricane Island, and the beach that we cleaned is right here. And this isn't even all the garbage that we collected. But you can see there's a lot of fishing related debris. And then in these trash bags are the coffee cups, the bleach bottles, so on and so forth that we're also finding. Now, if I bring this a little bit closer to home, over here on the right, These are all plastic fragments and parts of styrofoam coffee cups that I picked up on a casual half-hour stroll along shore of Palisades Reservoir. Okay. Down here on the lower left, we're getting smaller and smaller with our bits of plastic. These are hands of my students back in 2014. I had a number of sand samples that were sent to me from the shores of Lake Champlain in Vermont, which is where I grew up. And they're sorting through all the plastic debris that was in those just gallon bag sized sand samples. Over here in the middle, this is a famous photograph by a man named Marcus Erickson. He's one of the pioneer researchers in this field. He founds Five Gyres organization off the West Coast, and has sailed to the Pacific Gyre a number of different times and all over the world on expeditions looking at marine debris. And again, this penny here is for scale. So we're getting smaller and smaller and smaller with our plastic debris. Finally, over here on the right, this was sent to me from a colleague and a friend of mine from the Rozalia project, Rachel Miller. This is a picture of fibers she collected from her washing machine effluent after washing a fleece garment. So you might be sitting in your chairs thinking, well, okay, so there's not these large objects of debris floating around in the ocean, okay? They're getting smaller and smaller. There's no eyesore. You know, if an animal eats it, won't they just poop it out, right? So that might be what you're thinking, but what we're learning more and more is that these microplastics are actually causing a much larger problem than macroplastics, the larger objects. So much so that, in fact, United Nations Environmental Program last year just named microplastics as one of the world's most emerging environmental contaminants or concerns. Okay. So I'm going to take a minute and define microplastics for you. The most widely accepted definition of microplastics is any plastic fragment that is smaller than 5 millimeters in size. So you're going to have to use your imagination a little bit and get your fingers up there. But as we learn more and more about microplastics and the potential ill effects that they have, there are conversations about changing the categories. There's conversations about introducing another category called mesoplastics. Mesoplastics would then be 0.5 millimeter to 5 millimeters in size. Microplastics, it's suggested we should call any plastic fragment that is 0.5 millimeter, so 500 microns, down to 50 microns. And then we have to introduce nanoplastics, anything less than 50 microns. So as we learn more and more, we're finding smaller and smaller bits of plastic. And there's two general categories for plastic sources. We call one the primary sources, or these are your pre-production plastics. And some examples of that we call nurdles or resin pellets and microbeads. Now, I have an example of some nurdles or plastic pellets here that I'm going to pass around. And if you can just kind of hand it to the closest person next to you, and eventually it'll end up in the back. This is made of glass, so just be careful when you pass it around so you don't drop it. But what nurdles are, thank you, this is kind of how plastics are shipped around before they're turned into a plastic product, okay, like a toy. And so what you can do with these pellets is you can pour them into a mold, you melt it down, and when it hardens, you have the form of the product that you want to make. So that's one primary source of plastics. A second is microbeads. So microbeads are produced to be small. That's their purpose. And they are purposefully put into exfoliant body scrubs, toothpastes, sunscreens, lip gloss, eyeshadow, and I'm not really sure where the eyeshadow, the plastic in the eyeshadow comes into play, but they are purposely put into these products. And they might enhance viscosity of the product, they might help form a plastic film of the product, or they might act as a binding agent. So they do kind of have these purposes. How many of you have heard of microbeads recently? Yes, okay, so again, in the news, right? And the Microbeads-Free Waters Act of 2017 was just signed by President Obama. So hopefully this primary source of microplastics and it entering our oceans will start to decline. But what happens is our wastewater treatment facilities are not designed to capture all of them. So once they go down the drain, they can then end up in our rivers and our waterways. A secondary source of microplastics is a more obvious one, and that's just it results from that littered water bottle that gets thrown out into a water source. And as you get mechanical degradation, so ice and water freezing and melting and freezing and melting, wave activity, temperature fluctuations, UV light, it breaks down into smaller and smaller fragments. I have another example of some are actually larger than 5 millimeters, but if you look in here, you'll see some plastics that are well on their way to being microplastics and even perhaps that smaller category. These were also collected at Palisades Reservoir. So another source of the secondary, or another example of the secondary sources are microfibers. And I mentioned those earlier. Again, they're shed from our clothing when we wash them in our washing machines. And again, wastewater treatment facilities are not designed to filter them out. They might filter some of them out, but not all of them. And there have been a few recent studies. One was funded by Patagonia, telling you the numbers of fibers that could be shed upon a washing. This is a new scientific study that's currently in press, where they break down the different types of fabric. So you get polyester cotton blend versus pure polyester, which is what our fleeces are, to acrylic. And so they get a range from, on the low end, just 137,951 fibers per wash, to a higher end, 728,789 fibers. That's a lot of plastic. And a study off the coast of Nova Scotia, where they were looking at plastic ingestion in mussels, found over 100,000 plastic fibers in one mussel alone. So I wouldn't want to eat that mussel. So that leads me to talk about what are some of the ecological consequences. And there's some obvious ones that we'll call the physical hazards, right? So the ingestion of these plastics. So typical results are that an animal will eat microplastics, and all of their physiological responses are in order, right? The stomach stretches, sends signals to the brain, you get a sense of satiation. However, they're not getting any nutrient gain from those plastic particles, so they end up starving to death. Up here on the left, these are all objects that were taken out of one northern fulmar that was found on a beach in Washington. Okay, and this was just within the past 3 years or so. A college student actually collected this data. In the middle here, this is a lanternfish caught by Marcus Erickson in the Pacific Gyre. You can see all these plastic fragments that were in the gut of that fish. And we're getting even smaller. This is a famous video that circulated, and this, of course, is just a photograph, by Matt Cole, who's at the University of Exeter. And he was studying whether or not plankton ingested these microplastics. And so he fluorescently stained some microspheres and found that the plankton were actually ingesting these microspheres. Down here in the middle, this is a photograph I took of a plastic particle I found in the stomach of an Atlantic herring, which is a really important species in the Gulf of Maine. It fuels the lobster baiting industry. It's also at the bottom of the food chain for a number of different species that feed on it. So these are some of the more obvious examples of the hazards, just simply ingestion. If an animal happens to ingest a fragment that has a jagged edge, it could cause a laceration, okay, as well, or it could cause blockage, where nothing then can move through the gastrointestinal tract. This is a table here that's actually already outdated. So this was published in 2015, looking through the published literature to try to get an estimate of how many species we know of have ingested some form of plastic, but that number is now up around 600. Just this week, there were publications on a giant squid and microplastic ingestion in the squid, and then some organisms at the depths, the deepest part of the ocean, have also been known to ingest microplastics. One less obvious hazard here with microplastic ingestion is hitchhiking. So this is a photograph that I took. This is a plastic fragment I just found on a beach in Oregon this summer, and on it is a snail. Okay. So hitchhiking, we're just starting to learn about the consequences or potential consequences of this. But invasive species could adhere to these plastic fragments, and as that fragment moves around the ocean, it could then colonize in areas where typically they wouldn't be found to do so. And this holds true for pathogenic bacteria as well. The less known hazards are the chemical hazards, and we're still trying to put together all the complicated pieces of this puzzle. But essentially, if this is a fragment of plastic, okay, there's a lot of chemical additives that go into virgin plastics during production. So for example, fire retardants, so that your product is fire resistant. Triclocans, so that it is antibiotic or antibacterial resistant. Triclocan was just banned by the FDA, by the way. So plasticizers, whitening agents, a number of chemicals can go into those plastics during production, and some of them can be quite toxic. But then we have these other chemicals that, if they're already in the water source, they're attracted to the plastic in the water, and they essentially stick to that plastic, and the proper term is called adsorption. Okay, so this is a famous figure by Chelsea Rockman, who's a scientist that's really pioneering this field and looking at what are the toxicology repercussions for ingesting plastic beyond just a laceration or causing starvation. And there's a lot of text on this slide. But what I want to show you is these are very recent lab studies that are looking at this. They're looking at what are the effects on various organisms when they do feed on microplastics that don't have these chemical pollutants added to them, and when they do. And what they're finding is pretty astounding. Hepatic stress, so stress of the liver, endocrine disruption, altered feeding behavior, and reduced energy reserves, so reduced lipid storage, which is pretty important if the lugworm is at the bottom of the food chain, and animals depend on that lipid source for energy. Oxidative stress, mortality, here's an interesting one. Endocytosis of the plastics into the digestive cells, so that's at a cellular level. Reduced growth rate, activity, predatory response, and in this one, these perch, they actually ended up showing a preference for eating the plastic. Okay. Then we have decreased fecundity, decreased ingestion, mortality, and a number of different organisms. We have the Japanese medaka, which is a small fish, lugworms, crabs, mussels, okay? So after some of these lab studies were conducted, the question still remained, well, okay, so what happens then if another organism then ingests the organism that ate all those plastics? Okay, so the trophic level transfer, and what is the potential for bioaccumulation? And that's kind of where the research is going now. One recent study did show that when they fed blue mussels microplastic particles, they found chemicals getting taken up into the mussel's tissues. And then they fed the crabs these mussels, and they were finding the chemicals then appearing in the crab tissues as well. So that's kind of the first study really looking at this issue of bioaccumulation. And this is kind of where it hits home, because we all rely on the oceans. Many of us eat fish. We're at the top of the food chain. Okay, so that's kind of the unanswered question. We really don't know what the repercussions could be for human health. But we do know that there are repercussions in our species that ingest plastics. And so this is a really good summary photograph Over here on the left, we have our kind of our secondary sources and primary sources of plastics. We have weathering, biofouling. The plastics can work their way through the water column. Okay, in the middle here, we have our chemical adsorption of pollutants. And then over here on the right, the potential for bioaccumulation up the food chain. So up until now, everything that I've told you is basically about species in our oceans and marine debris in our oceans. We really do not know much about freshwater systems. And it really hasn't been until the past 5 years that we've really started to look at that. And most of the research has taken place over in Europe, with the exception of Mongolia and Lake Victoria in Africa. So this is just kind of a list of a number of locations where microplastic contamination has been confirmed. If we take it home to the US and Canada, we know there's microplastic pollution. In fact, a significant amount of microplastic pollution in the Great Lakes. We now know, as of last month, that there's microplastic pollution in the tributaries that empty into the Great Lakes. The USGS just completed a study and published it in September. We know about some rivers near Los Angeles and the St. Lawrence River. That's about it. So there is a need to understand the dynamics of microplastic pollution in our freshwater systems. Our rivers are linear, okay? We don't have the gyres that we do in the oceans. Or do we? Do we have accumulation zones in our rivers? Do we have accumulation zones in our eddies, for example? Or what about dams and reservoirs? How does that play into where microplastics might accumulate in our freshwater systems? And if they are accumulating, what effect, if any, will they have on our freshwater organisms? So that leads me to describe to you a study that I'm currently working on with students here more locally. And we're looking at microplastics in the Snake River. And the Snake River is a really unique river in that its headwaters start over here in the largest, most intact ecosystem of the lower 48, okay? It is over 1,000 miles long. It travels through 4 states. And it travels through a variety of different landscapes. We get national parks, we get wild and scenic, we get heavy agriculture down here. There's over a dozen dams on the Snake River, and then it is the largest and most important tributary for the Columbia River. So we had some initial samples back in 2015. They were simple, just grab samples. One of my students collected them. 8 out of 11 we found sign of microplastics. And over here I have 2 examples. Now I call them just anthropogenic fibers because, as I talk about in a minute, you need to confirm, and you need to use a special form of microscopy to confirm that what you're looking at is indeed plastic. But these 2 fibers fit all the visual criteria for that. A consistent width all along, okay? A pretty homogeneous color, and no real internal structures, okay? So I call those anthropogenic fibers. Over here on the left, this sample we took near Heisey Hot Springs over in Idaho. And we did confirm this blue fragment here to be a copolymer. And down here we see this perfectly round red bead. Unfortunately, we could not get a confirmation on this because what that involves is beaming a little laser light at the object. And unfortunately, after we did that, we had a hard time getting a really good reading, and then it melted. So back to the drawing board on that one, but it looks a lot like a microbead. So this past summer, in early August, my research assistant, Ellen Yeatman, who's here in the crowd, and I sampled, we decided to sample the entire length of the Snake River. And we collected water samples from pretty close to the headwaters up here. We did not hike all the way into the headwaters of the Snake, but right at the south boundary of Yellowstone National Park in a little bit. Every 50 river miles, all the way to the Columbia River, and then followed the Columbia River out to the Pacific Ocean. And these are pictures of some of our various sampling sites. So I'm not going to awe you and woo you with numbers and concrete data because we are still very much in the process of sorting through our samples. And I'm going to walk you through what that process entails so you can maybe feel some of the pain that we feel right now because it's a very tedious process. So at each site, we collected 2 samples. We collect just a 1,700 milliliter sample in a glass jar, and we built this pole that you attach the jar to get it as far away from our body as possible. A major concern with these studies is contamination risk, because there are fibers shedding off our clothing at all times. Okay. But we don't really feel that that tiny little sample of water at that fraction in time is very representative. So we can't really be all that confident that if we find a fiber, okay, yes, they're there. If we don't find a fiber, that they're not. So we also collect what we call volume-reduced samples. And this is when we use a plankton net. In this case, it's got a mesh size of 100 microns. So that means that the pores, okay, in the net are 100 microns. So we can only capture anything larger than that, because anything smaller is going to move right through the holes in the net. And here it is here. You can see it's a round net. And we attach a flow meter to it in the mouth of the net so that we can adequately gauge what volume we're actually sampling. And by using this method, we can sample over 1,000 liters as opposed to just 1,700 mils. So it gives us a little bit more of a representative sample. And depending on how we collect the sample depends on the type of site. So sometimes we can easily wade out into the river. Other times we luckily caught rides on motorboats with game and fish, for example. Or if there's a bridge that was safe enough and low enough, we could drop our net down off the bridge to collect our samples. Well, because we have these 2 different sampling techniques, we also have 2 different methodologies that we need to follow once we get back to the lab. We do all of our sampling processing in this laminar flow cabinet here. It's also called a clean air cabinet. And what that does is it sucks the air from the room through a HEPA filter and then blows it back out at you so that hopefully in that chamber you have no dust or fibrous particles that might be in the air. And what we're doing with our grab samples is we just pour the whole sample into this beaker. And down here is a filter paper. And it has tiny little holes in it that are size of 0.45 microns. Okay, so there's 1,000 microns in 1 millimeter, just to give you an idea of how small it really is. And so all the water gets sucked through a vacuum pump, and then everything that's floating in the water ends up on the filter paper. And then we can take it off and start to examine it under a microscope. Our volume-reduced samples, though, are a bit more complicated. As you can see me looking fairly perplexed here in this photograph. This is an example of one of our volume-reduced samples, and you can see all the material here floating on top. So if we just use that simple technique I just showed you, our filter papers would clog pretty quickly. And so, not to mention it'd be really hard to dig through and separate out, well, what is plastic and what is not plastic, because that's really challenging. So we send that sample through a stainless steel sieve. We then will vacuum filter this. But this we'll put in a drying oven, and we dry it in the oven 90 degrees Celsius for 24 hours. And then we have to apply a digest to it. So our goal is to break down the biological material. And we adopted a pretty widely used method. And it's also adopted by NOAA. So they use it. The study that was just published looking at the tributaries in the Great Lakes also use this method. But some scientists use acid digest as well to break down the biological material. In this case, we use a 30% hydrogen peroxide with a catalyst added to it. You don't want 30% hydrogen peroxide on your finger. What you put on your finger if you get a cut is 3%. Okay. But we expose it to heat. You can see the bubbling activity. That is it working. It is digesting the biological material for us. But then we have this kind of sludge here, and we still need to figure out what's plastic and what's not. So what we do is we add this to a highly saturated salt solution, right? Because some plastics are not very dense, and they float, right? It's kind of like the Great Salt Lake. If you go swim in the Great Salt Lake, you can float, okay? So we have some of these plastics up here with pretty low densities. And so we're adding them to a mixture of sodium chloride, which is a density of 1.15 grams per mil. And we're hoping to float out some of these plastics. But we then do it again. So we put it in here, we let it sit for about an hour or so, then we drain off the sediment, and we add that sediment then to sodium iodide. And that's a more dense, higher density solution, and so we hope to float out even some of these more dense plastics here. Okay, so that's kind of walking you through the methodology. And this is the difference and why we do this. So on the left, we have a photograph taken through a microscope of a sample that we did not use hydrogen peroxide. And you can see all of this extra material in here makes it really difficult to find plastic. But over here, after using a wet peroxide oxidation, a lot fewer particles are in this slide. But it's still really difficult to tell what you're looking at. And so just in the past month or so, we've started using a fluorescence illuminator with our microscope. And so up on the top row, these are all images of particles, or our filter paper, under just a light microscope. And down here, these are the same images, but they are now fluoresced. Okay? So for example, over here, it doesn't look like much, but once I add some blue light to it and illuminate it, you can see some of these items are fluorescing. Now I should mention that some biological material fluoresces, and not all plastic fluoresces. So I can't say that, oh, that's all plastic. That's not true. But it does help us tremendously when we're trying to dig through our samples to determine what is plastic and what is not. This over here, for reference, this is confirmed plastic. This is the particle I found in that Atlantic herring gut. So this is kind of the behavior of that high-density polyethylene and how it's going to respond when you illuminate it with this type of light. And this is very similar and very suspicious looking. As I mentioned before, though, you cannot just base or determine if something's plastic by looking at it. You then need to apply a special type of microscopy to confirm. And there's two different ways you can do this. We choose to use Raman microscopes. And what Raman microscopy does is it's called a light scattering technique. So what it does is it beams a laser light at a molecule or a material. And that excites the photons, and it makes that molecule kind of jiggle around a bit. And as it jiggles, the light reflects away. And the machine then can measure that scattering pattern. And every material has a specific, what we call, fingerprint. And so down here, these are the fingerprints of polyethylene, a low density and a high density. A high density polyethylene is what your laundry detergent bottles are made of, for example. And this is that fragment from the fish gut. And when we shine the laser light at it, you can see how the fingerprint is very similar to both of these. Now, I am not a specialist in Raman microscopy. Luckily, Dr. Keith Karen with Snowy Range Instruments, a former faculty member at University of Wyoming, has offered to help. And so he helped us. This is his Raman microscope. This is one of my former research assistants, Greg Van Gilder. That's that red microbead that we were trying to get a fingerprint on. But you can see how this works, and Dr. Keith Karen confirms that, I think it's actually high-density polyethylene that you found in that herring fish gut. So after we process all of our samples using the density separation techniques and sort through them, we will then bring them all the way down to Laramie and spend a lot of time using this Raman microscope here trying to confirm whether or not we're looking at plastic. And this is an essential step. A number of studies have showed that plastics that were identified just by visual criteria under the microscope, actually 70% of them are misidentified. So it's really important that we provide accurate information and really positively identify these as plastics. So in summary, We know that microplastics are ubiquitous in marine environments. Lab studies now show that there is harm to organisms that ingest them, that they are indeed available for trophic level transfer, but we need more studies, we need more research. Okay, we need more research to understand the dynamics of microplastics in our freshwater systems. And finally, we need to reduce our plastic footprint. We need to reduce, reuse, recycle, and refuse some plastic items that we can certainly live without. So hopefully I have time for some questions, and if there are any.
Thank you. Based on the study that Patagonia did, has Patagonia or other major clothing manufacturers since done anything to change their manufacturing process and/or use of materials?
Good question, good question. Patagonia is the leading company in that. I know a number of other clothing companies have not actually responded to requests to speak with people about those types of results. I'm not sure to date of any new fabrics that Patagonia is choosing to make, but they do advocate their repair policy. Instead of buying a new jacket, repair it, for example. But other than that, I'm not sure of any current designs that they have to undergo that, other than to continue funding the research.
I actually have two. The first one, for like corn plastics and other plant-based plastics, are they having the same kind of effect in terms of, animals eating them and then, starving to death or floating around, those sorts of things?
That's a really good question, and there's not any research right now to date looking at that as far as the chemical properties of those plant-based. But lacking some of the additives that are needed to make that plastic, I would suspect that the consequences are not as great.
And then my other question was about the bacteria that they're trying to help digest plastics. What kind of effect is that having in terms of reducing the impact?
That's another really good question. And they have been successful at finding bacteria that do digest plastics, but then you have to think about how are you going to get those bacteria into the ocean system? And then on the scale, right, the type of scale, the problem that we have, how is that possible? And then what are the consequences of introducing a bacteria to those natural systems to see how it would work? So it's really interesting, but challenging too.
Do we know anything about the interaction between ocean acidification and microplastics?
Good question, too. I've often wondered that about both temperature and ocean acidification. And to date, no studies have looked at that. Yeah, they've tried to start collecting some data, just looking at how does the pH change between higher densities of plastics in the ocean and not. But so far, there haven't been any results looking at that. Yeah.
Sort of briefly touched on how some chemicals are attracted to plastic. Mm-hmm. Is there any way to, in a sense, create, and I don't know what it is or would be, something that would be like a magnet that would start to collect the plastic so we could bring it to one place and dispose of it properly?
Good question. There have been a couple designs out there. One, some of you may have heard of, a 19-year-old by the name of Boyan Slat developed this kind of huge anchored boom system where he would try to take advantage of the ocean currents and then try to filter out the plastic objects. But unfortunately, the consequences of doing that outweigh the benefits a little bit. There's some controversy involved with that, but the amount of plankton that you would also have to capture as you're trying to filter out these small microscopic pieces of plastic is huge. There has been some developments in washing machines where they try to put objects in there that work like a magnet to try to attract those plastics to that. And actually, while you've asked the question, I'm going to show you the picture of another development here by the Rozalia project. This is what they call their microfiber catcher. It's currently in design right now, but their goal is to put it on the market next spring and to sell it with washing machines and then have individuals purchase it. And inside, there's kind of just a great surface area of plastics. And if plastics are attracted to plastics, and you could just throw this in your washing machine while you're doing laundry in hopes to filter out some of those fibers. That's one example of developments underway. And there's some other developments too. But as far as looking at the ocean and how we can clean it up, unfortunately, it's kind of an unfeasible process. Yeah.
When we look back, like the plastics have been around since the 50s, and I know you said you wouldn't eat that mussel, but I'm sure we all have eaten a mussel that's been completely plastic full. Or, you know, so it's, are studies being done about just the general vastness of this whole, you know, I wonder how old the plastics are that we've eaten or whatever, you know.
It's a really good point. And no, nobody has yet looked at the plastic particles that they do find in at least the species and tried to date it back. Is that kind of what you're, in a sense? Yeah, that would be really interesting to know how old those plastic particles are in the ecosystem. And then as far as the human component, a study published, I believe it was 2 years ago now in Science magazine, they looked at fish that you're buying right at the market and kind of trying to calculate how many of those fish species are actually making it to the table. But still, we're still in the infancy of looking at chemicals that are transferred to the actual tissues and then finding them in our tissues. And that's a really difficult study to do, but hopefully going in that direction.
With this gadget from Rozalia project, if you capture the plastics in this gadget, then what do you do with the waste that's in, with the plastic that's inside?
Good question. And their plan is that you would subscribe to this. So you would have your microfiber catcher. When it gets full, you'll mail it back to them. And they will clean it for you. And then you get a new one. What do they do? They then will hope to extract it and use it to produce recycled objects from the fibers. And I don't know what types of objects that is. So yeah, it's an example of kind of the innovation involved with this issue.
Are there any efforts to remove the macroplastics? Because sooner or later, they'll degrade and become microplastics.
Absolutely, yeah. There's... Oh, yes. Is there any effort to remove the macroplastics? Because eventually they will turn into microplastics. And there's World Ocean Cleanup Day. There's a number of beach cleanups and working on shorelines to do that. There's a lot more efforts kind of trying to look at the land sources. There's a neat water wheel in Baltimore, Maryland, for example, in the river, because rivers are an incredible source of the marine debris to the oceans. And it spins, and after a rainstorm, all this trash ends up in the river, and it's really neat to watch. It spins and captures all the larger pieces of debris. As far as out in the oceans, there are some boats that go out, like the Rozalia project, and clean up what they can. For example, when I was on board, the number of balloons that we found floating in the water, and we'd stop and get out our long net and try to pull it out of the water. So hopefully that answers your question a little bit, but you're right, that's a huge source. And even if we stopped all the input of plastics and debris now, we're still gonna see an increase because of that reaction of macro going to micro.
Yeah, a question about your two sampling techniques. How do they correlate, the grab sample versus the reduced volume?
Very good question, which is the reason why I did it, but we don't know yet. So I'm hoping to look at that, because it would be really amazing if we found that the 1.5 liter bottle correlated well to the larger net, because it would make sampling so much easier in the end and processing those samples. But we're not sure yet how that's going to correlate.
Okay.
Okay.
Thank you very much.
Paul.